Marissa. I am holding a microphone. My voice is not going to be any louder than it is right now. This is just for our recording. We do record all of our videos, all of our Saturday seminars, and we post them on our YouTube. So if you've missed any, or you want to share this with anyone, you can find those recordings on our YouTube page. But for those that don't know me, my name is Marissa Blackburn. I am the Environmental Education Manager here at Cape Fear River Watch. And so I thank you for coming out on Saturday morning to learn with us, especially if you joined us last night for our 30th anniversary birthday party and had a late night last night. So we're really excited to have you here, and we have a really great talk today. Before we get started, as always, I'm going to share a couple updates about Riverwatch. We do have a cleanup next Saturday, the 8th. Details are still TBD, so we will probably be sharing that on our social media on Monday. The following Thursday, June 13th, we have a pint night at Wrightsville Beach Brewery, so please join us for that. We are the Wrightsville Beach Brewery's nonprofit partner recipient for June, so that means we get a portion of proceeds from one of their signature beer sales. So again, that's Thursday the 13th. And then on Saturday, June 15th, we will be having our June paddle. We will be paddling Sharktooth Island and Eagles Island. And Angela Caldwell-Marshall will actually be leading that one instead of Kemp, and she is from Kayak Carolina. And our registration for that is still also going to be available very soon, but is not quite available yet. So just keep an eye out for our e-news and also our social media accounts for that. And that's all I have. So not a lot of Riverwatch updates this month. So I am excited now to introduce our speaker for today, Dr. Monica Rother. She is an associate professor in the Environmental Sciences Department at UNCW. She conducts research within the broad disciplines of forest ecology, biogeography, and dendrochronology, or tree ring science. She earned her PhD from the University of Colorado Boulder in 2015 and previously worked as a fire ecologist at Tall Timbers Research Station in Tallahassee, Florida. She teaches courses in tree and forest measurement, environmental conservation, and wildland fire ecology, among other topics. And if you want to learn more about her and her research after today's talk, she does have a Google site which you can visit. And so with that, I'm going to turn it on over to you, Monica. Thank you. And Nick, can you turn off the lights?
Benefits for society and the environment. So of course, storm buffering and flood mitigation as well. As these ghost forests form, we have marshes instead of forested wetlands, right? We have treeless landscapes, and those they have their own value, but they don't provide the same services as the forested ecosystem. So we think about carbon storage, erosion control, preventing the sediments from washing away, filtration to improve water quality, all sorts of things. And there's some work, including in our region, that the areas where these ghost forests are forming, it tends to be affecting disadvantaged communities disproportionately. So that's what I mean by environmental justice. So lower income and BIPOC communities being affected by this problem disproportionately. Okay, so here's a look at some of our local sea level trends. So we know that globally sea level is rising. It has done so more here than the global average, and that's that extra effect of dredging. So we would expect a little bit of sea level rise, but we're seeing a lot more related to dredging. So here's some of the historical data. Now, dredging of the Cape Fear River, there's kind of always some ongoing maintenance dredging, as it's called. So these are just the big events where they actually went in to try to make it much deeper, right? So you see the dredging going on kind of continuously, but these are the biggest events. So the most recent large event went on from 2000 to 2005. You can see the red line there, kind of the spatial scale of that dredging, deepening things from 42 to 44 feet from the previous 38. And we see, as you'll see as I continue on in this presentation, 1970s and 2000s, we do see kind of revealing what we'll find here early, but we see shifts that correspond with those big events in the trees and the forams. I'll talk about that in a minute. Okay, so I mentioned the tree rings and the sediments. But specifically when I'm talking about the sediments, we're talking about foraminifera. And I'm going to call them forams from now on because it's such a mouthful, I can hardly get it out. But this is, again, what Dr. Hawks looks at a lot. And what's really useful, and the reason we were like, we should come together, is that they can be a great indicator of salt. So we wanted to pair these proxies, as they're called. And what that means is, you know, a thermometer is a direct way to measure temperature. But tree rings or microbiota of whatever kind, it can give us an indirect way to look back in the past for time periods when we weren't necessarily taking direct measurements. So with tree rings, as some of you may know, that's how we know, it's ice cores as well, but we know a lot about the long history of temperature on Earth from trees, right? So we can use tree rings and other proxies, as they're called, to learn about historical environments. So dendrochronology is the mouthful word for tree rings, tree ring science. So, you know, it's got tree in there and then chronology for the study of time. But you can also use it for spatial patterns, which we do in this study. So I'll go ahead and pass around my increment borer, although I'm going to close it before I do that. But this is the tool. This is a big one. They're often skinnier than this, but it's a somewhat wide one because we wanted to make sure to get a big enough sample that we could see what we needed to do. So here's the extractor, or spoon as it's sometimes called. But what these do, these increment borers, well, has anyone used one before? Nice. Yeah, my students. I got a couple students in the room. Great. And some others. Yeah. So this allows us to take a pencil thin, maybe a little thicker than a pencil with this big one, sample from the tree. It does create a very localized injury, but it does not leave long-term damage on the tree. So there have been many long-term studies where they go back after you've cored the trees, and the trees are definitely okay with this little poke, right? And you think about all the things that birds do and all the kinds of injuries that trees can endure, they do okay. So it clips together like this, and you twist it into the tree, and then you put in the extractor and you pull out your core. So that's kind of the field work portion of the tree ring work. And my colleague, Dr. Hawks, she has her own borers to get down into the mud, into the sediments, to collect her samples. So keep this closed unless you want to gently open it and close it again if you feel like you know what you're doing, but I'll pass it around. This is also a murder weapon, you'll see, it's pretty heavy. And then I'm kind of jumping the chase, but it's fun. I'll pass around one of our cores, and it's dark in here, but try to see the individual rings. This turns out to be, this is our oldest tree out of the, I don't remember how many total trees that we cored, 20 to 30. It's in the presentation. So this is the oldest one, and I'll talk more about that as it goes on. Okay, so the forams, again, I mentioned that they're really useful for understanding things about salt and salinity, and that's because they only occur in salty environments. But there's a gradient. There's many different types of the forams, and some of them like it really salty, some of them will occur in brackish environments. So this is a way where we can look at, through the sediments, through time and space. Again, both these proxies let us look, how are things different at place A and B, and how did it change through time, which is really cool. So the forams help us get at whether it's really salt, essentially, that is related to this. So just a really simple map to orient you guys to our study area. We're downtown right now, just south of the airport. Essentially, that creek there is Smith Creek. Who's been on Smith Creek? Wow, this is the audience for this talk. I love it. Yeah, so that's where we first started this study. And when Andrea came to me, Dr. Hawks, my collaborator, we're like, thinking of this as kind of a pilot study. We didn't know if we'd find anything interesting, but we did, as I'll share here soon. And so I hope to continue this research and grow this research in the coming years. Okay, so for those of you who have been on it, you know that there is a tree mortality gradient. And what I mean by that is that you go from closer to the Cape Fear River at the mouth, all dead trees, nothing but ghost forests. But as you move upstream along Smith Creek, then it kind of becomes a mixture, and eventually you get to a point where you no longer have those dead trees, right? You no longer have those snags. So this creates a nice opportunity to kind of study spatially what might be changing along that gradient. We did already identify and kind of study the salinity gradient that goes along with it. So this is called a castaway, and essentially on that previous map, you see those little, they're small, but you see those white points. Those are where we sampled with the castaway to kind of figure out how salt is changing along the Smith Creek. But we're also now, they've just recently been installed some long-term sensors that are going to continue to monitor salinity nonstop, and also water level, because we want to understand better the relationship between those two. So I didn't really say that earlier, but the saltwater intrusion problem and the connection to dredging is that when things are deeper, then that allows more of the salt to get in, right? And it increases the tidal amplitude. So it also affects water levels. They're paired. So we ended up starting with just these two sites. So yes?
So with your locations, did you take salinity at different times with different tides?
Yes, I'll show you. Yes, good question. So he wondered whether or not we sampled salinity on more than one day. We did, but 3 days total, and that's why we're putting in the permanent sensors, because we realize that that's inadequate because it changes so massively. Yeah. Okay, so here's our two study sites, the ghost forest site and the healthy or healthier site, the less salty site, the brackish site, very slightly brackish upstream. Oh, well, there's the data right there that you were asking about. So you see those 3 lines, that's the salinity gradient on 3 different days. So you can see there's variability. The May 22 line, which is the red line, is flatter, right? So the gradient wasn't as strong compared to our other 2 dates. So this indicates that in general, it's definitely going to be saltier on one end and less on the other, but if we really want to, understand it, that long-term continuous monitoring is necessary. Did that make its way back too? Okay, I'm gonna pass it back. I know you guys want to look at this. Who's next? Everybody's good? Okay. Maybe it did. Okay, so into the tree ring part of it, again, we had our 2 sites. So in our low salinity or healthier of the 2 sites, we cored 17 live trees. And we take 2 cores per tree because it doesn't always work out. And with some of these, they were hollow inside, so we didn't necessarily get all of the rings, but we did our best. And the bigger the bore, this is the first time that I worked with a wider bore, the harder it is to core. So this took a lot of work, and especially coring in these wet conditions. Okay, so then the high salinity site is in the intermediate area where it has a mixture of live and dead trees. We didn't want to work just like in the full-on ghost forest because they have been dead for so long that you're missing the outermost wood. So we want to know, what's happening as these ghost forests are forming. So that's why we really wanted to focus on the live trees or maybe trees that just died. Because as you guys kind of know, as trees die, again, you could lose 20, 30 rings just kind of shed off as that deteriorates. You also can't really core it anymore. The wood becomes junky. So we focused again on this mixed site where the trees were dying actively, and then a healthy site that was kind of almost like a control site. So we take the cores back to the lab and let them dry out a little bit so they'll do the shrinking that they're going to do, glue them down to little wood bores or little wood mounts. You see the core that I passed around. Sand them so that we can see the rings nicely. And then This work used to always just be done under a microscope, but now with technology, it's quite nice. Early in my career, it was all microscope, and now I'm all computer, and I'm a fan of the computer stuff because we can scan it, and then we can look at it on a nice big monitor, very comfortable. And it also allows you to do things that you can't really do very easily under a microscope, like draw on the wood, you know, and share with collaborators and do all sorts of other things. So here you can see one of our cores, and I'm essentially counting these rings back through time. But it's more than just ring counting. Now, tree ring science, I always say this, it's not rocket science. It's not like the craziest thing ever. But you can't just count the rings. And that's because there are all these different ring anomalies, as they're called, that make it where your count's going to be a little off if that's all you do. So there can be missing rings in really harsh years where, at least for the part of wood that you have, there's nothing there. Or there can be these double rings or false rings where the tree thought it was kind of done growing or it should start shutting down, but then it starts again. So there's a lot of things that complicate it. So that's why we use this cross-dating process. And what that means is we find the shared pattern among many different trees. And why are some rings in general, what does that mean, a wide ring versus a narrow ring? Yeah, something's good versus not, right? So when conditions are favorable, and usually it is climate driven, like maybe precipitation, whatever, it could be temperature with some species in some locations, we grow more. When conditions are less favorable, we grow less, right? But we can use that pattern to then match up the different trees. So they'll have these shared, you know, here you can see 19, very narrow ring. That's likely to be shared among the other trees. It won't look exactly like that because trees are individuals, they do their own thing. But we do this pattern matching first visually, and then we use correlation analysis because this is actually capturing the exact width. So the rings are being measured here as well. And we can do correlation analysis type pattern matching to verify our dating. So that ensures that every single ring is dated correctly and it's not just a rough estimate. So here's another look at cross-dating, you know, the visual process. So I know it's a bit hard to see, but essentially what the white arrows are connecting the same year across these different samples. And you start by kind of focusing on the narrow rings usually in tree ring analysis, and you'll get your kind of marker rings like, oh, 1902 is supposed to be narrow, 1876, and that helps you make sure that you're kind of matching them up. And then again, we verify that statistically through correlation analysis. And then we can develop the shared pattern of growth. So I won't go into the weeds here, but essentially you don't want to look at just one tree because an individual tree, maybe it has some disease going on, maybe competition, some other thing. But when we look at the shared pattern of the trees from the site, what are they generally doing? So that's what you see here, the ups. The trees, most of the trees are growing well. The downs, most of the trees are not growing well through time. This is just an example. This isn't our results from this study. This is just an example chronology. But we built chronologies for our two sites. So a chronology for the healthy site and a chronology for the in process of ghost forest site.
So in the correlation analysis, how much of a local area are you talking about? A few meters or a few kilometers?
For the chronology?
Yeah, for the correlation you're building on.
Like how far spaced are the trees?
Right.
Is that what you're asking? Usually, it can vary, but usually like no more than like half a mile apart. But sometimes you go through larger areas. Yeah, so it's pretty local. But then we can combine the chronologies to build these more regional chronologies if we want to look at top-down signals like really temperature and if we want to really look at climate. But for this study, it's better to be localized with the questions we're asking. That's a good question. So you can see too, some of the spatial spread is different between our two sites, but both of them are tight enough where, again, we don't expect a lot to be different at the individual site because they're at least close enough that we're not going to expect rainfall to be different at these two sites or within the individual site. But part of that is because in this area here, most of the trees are dead, so we had to cover a wider spread to just find enough trees to core at our salty site. So I'll skip kind of some of the statistics there, but that's some information about our chronology. Okay, so moving into some of the meat of the results, we definitely found in green here is the trees at the healthy site. The blue is the trees at the ghost forest site. And you can see tree age on the y-axis, which site has older trees.
Healthier?
The healthier site, the upstream site. So what we think is going on, and other parts of our results support this too, is that a lot of the oldest trees closer to the mouth of the river have already died, and that those older trees are more vulnerable to environmental change and stressors, which is true in general in other contexts as well. So you also start to see these trees are old, right? It's sad to think that trees that are several hundred years old, in some cases our oldest tree was 800 years old, which is kind of the oldest outside of the Three Sisters area, definitely the oldest found in these tidal forested wetlands here locally so far. And we didn't even core that many trees, and one turned out to be 800 years old. Now it's currently in a safe setting, but as I'll get into, it's not actually, right? And as you guys probably know, more dredging is planned. But yeah, so even at the ghost forest site, a couple of the trees were 400 or older, and they're very vulnerable. Most of the trees in the area have already died. So here is the chronology for the salty site. So again, the ups, just focus on the top part of the graph. The ups are good years, the downs are poorer years. So if you see that line, when we dip below that, that means tree growth is lower than average. And here are those last two big major dredging events, 1970 and 2000 through 2005. And you see how that shared signal really takes a turn downwards, first at 1970, and then it continues to do so to the present. So we really don't expect, unfortunately, this is sad to share, but that the trees at that site will continue to live for that much longer, because they are very stressed currently and they're just kind of barely hanging on. Bringing back up that data. So again, 1970 being highlighted there, and then the 2000 moving into 2005, that was a multi-year project, is the second red line. This is another way of looking at that. So instead of looking at as much of the shared signal, on the y-axis there are the individual trees from the salty site. So SMC 119 was one tree. So each one of these lines is a tree. And what these events are here are suppression events. And that means that the tree was growing minimally for several years at least. The red is a very severe suppression event where the tree was barely growing at all, whereas the blue is just slightly narrower rings than usual. But you can see the temporal, so it's not just 1 year, right? These span several years. And what do you notice from the left versus the right side? More. The trees are stressed out. It's 1970 again. Remember, there was dredging before that, but thinking about 1970, 2000, those big dredging events. One more way of looking at that. So here is, instead of just what the trees are doing on average, what percentage of the trees are stressed. So again, these are just important to kind of do more than one analysis to make sure that we're seeing what we think we're seeing. So these are all very similar, but just a slightly different way of looking at the data. And here again, we're looking at the percentage of trees at each site that is stressed. So the low salinity site on the top versus our ghost forest site on the bottom. And we see many more, those black dots indicating suppression years in this case, where 75% or more of the trees are stressed. So this is telling us lots of different things, but one thing that I haven't emphasized yet is that this is telling us that the trees don't just suddenly die. They're not just doing great and then suddenly keel over, right? They are trying their best for extended periods of time, and that at a certain point it becomes too much. And we'll get into that a little bit more too. So the becomes too much can be related to other stressors. So essentially trees, they'll store a little bit of reserves to help them in the next year. But when trees are stressed, they don't do as much of that. So they just kind of do what they can to survive, and then they don't have that backup energy for the next year. So then if the next year is also hard, they're really hit back by that. And what you're seeing here, just in a nutshell, is that the trees that are in the ghost forest area that are still alive are very sensitive to climate. So there's much more correlation. The lowercase months are the previous year's climate, because that affects the reserves that I just mentioned. And the caps locks here are current year. And you see we have, especially with current year being what's different between the two sites, that if it's dry, that's really going to affect how the trees are growing. If it's wet, that's really going to make an impact. So the trees are more sensitive to climate when they're stressed out, right? Which makes sense. So maybe what actually gets the trees in the end isn't the salt exactly, it's that the salt has already stressed them to such a degree that then when an unfavorable year comes along, they're not prepared to endure it. Okay, so moving into the foram part. So this was to help us get better at, is it definitely the salt? Now we expect that because the spatial scale of our study, again, we don't expect things to be massively different between our two sites in terms of the climate they're experiencing, the local environmental conditions they're growing in, except for that. So that's what we suspect, of course, and that it corresponds with the dredging is pretty compelling, but this can help us really nail down the salinity piece. So first, forams have been used as a proxy for salinity in the past, but we wanted to test that. So first we just moved along our gradient where we had done the castaways and sampled the very top part of the sediment that had formed recently to see what happens along the salinity gradient. So again, all we're doing is, we know it's saltier, that way versus this way. Do the forams change just in the very top part of the sediment? Let's test and make sure the proxy is adequate. And we do see that. So we see, again, forams only occur in salty or brackish conditions. They're only at those stations closer to the mouth of the river, and then they disappear. And different species do slightly different things, so we can dig in deeper. But this was convincing that they'll be useful for this study, essentially. So then we went back to our two sites and dug deeper. I usually don't talk about sediment, so that was fun. To see what happened through time at the two sites. So we know that that first thing was just like the top level, but what happens as you go deeper into the mud, you're getting back in time, right, for when those sediments were laid down historically. So the top is more recent years, down deeper is moving you back in time. Very similar to tree rings, as you can see. So there's kind of some imagery to support what I was just saying about the top having the younger sediments, deeper down having older sediments. And then with tree rings, we don't have to use these methods as much, although sometimes we do. But we need, in this case, and these results are the last piece that aren't quite in, but in order to really know what time goes along with the sediments, we often rely on radioisotopes. So in this case, we're using cesium-137 to try to figure out, at least for a point along the sediments, what time was that? What year was that? So we're still kind of waiting for that part of understanding better how much, every 2 centimeters, how far back in time are you going? So at our healthy sites, we don't have forams, even all the way back. So that tells us that through time, and again, we're still needing more results to understand how much time we've captured, but that this has been a freshwater, or at least essentially fresh, maybe a little bit brackish, through time. It is today, and it was through time. So we don't see the forams there. We do see thecamebans, which are a freshwater, a different species that is only in freshwater. So those are there, supporting that same conclusion. But when we look at the salty site, we see that it wasn't always salty. So that's important too, right? So even these are tidal systems, but generally they're supposed to be mostly fresh water. And we see that where the ghost forests are, if we dig down deep enough, those forams disappear. So that means it used to be fresh there, right? So it wasn't always salty at our saltier site. So I know it's a lot, and I'm not going to go into the weeds, but these are the forams here, and so they're only in the top part of the core, and they're absent at the bottom. And then our freshwater, in gray, our freshwater species are further down. Yes?
You've shown us the pictures of what these little critters look like, but do they fossilize or something? How do you find them when they're really old? It would look like to me that when they died, they just go away. You find their carcasses, or?
Yeah. I don't know the answer because I'm the tree ring person, but I know they're there. So I know that Kendra looked under a microscope and counted them.
Like a shell, like a seashell?
Yes.
Are they diatoms? Because they have silica
They're
walls that stay. I did a study on a lake core using diatoms to try to.
I think these are different, but maybe it's similar in some ways. I'd have to put you, I can give, if you want your question answers, I can definitely get them for you. But again, this is out of my wheelhouse. This is the first time that I've worked with this at all, so I'm not totally sure. They're single cell organisms, so they're very tiny. But yeah.
So in the analogy to the tree rings, you have different depths for different years. You see that different depth for different year correlations in the board and the cores?
Yes, but it's not annual resolution like that, where it's just like, here's the next layer. So it's a course, you can go further back in time with sediments, which is cool. But you do things like with the cesium, it just gives you a benchmark, and then, yeah, exactly. Oops, I keep forgetting. The pointer works, but the arrow doesn't. Okay, so how am I doing on time? Anybody? Okay, good. Okay, good. Okay, so implications for this. This is where we are so far. Again, this is new research. We're working on getting it published now, but hopefully it'll be part of bigger studies. But we do think that the dredging events are what have caused the suppression in the trees, and that ultimately is what leads to the ghost forests. So the trees start growing less and less, and then they're less equipped to survive under those conditions. So currently, based on where the chronology has taken a turn, starting 1970 and continuing to today, these trees at our intermediate site, at that ghost forest site, are now kind of in permanently stressful conditions, and we're not expecting it to go back up based on decades essentially of suppression so far. And then, as I mentioned earlier in my presentation, some of our interest, we're definitely interested in the dredging and the problems that it causes, but also seeing this as a little bit of a window of what we might expect as sea level rise continues to affect our coasts. And so dredging, I know people in this room probably know more about it than I do in terms of the politics of it, but there are plans in place to continue dredging. So to bump now, it's a pretty big jump from 42 to 47 feet. They're wanting to do that so that they can bring bigger ships and stay competitive, supposedly, with the other ports. So I'm not 100% sure on exactly when they think this might start, but it's still in the works and doesn't seem to have big brakes on it. Does everybody agree with that assessment? Yeah. So when that happens, probably, and maybe even before, the remaining trees at our intermediate site will likely not be in good shape. I'm not sure whether the ones further upstream, if that's going to be enough to start to affect those trees. We'll have to see. I guess time will tell. Yes?
I got an email this morning. There's a new public comment period coming up on all this very soon this month.
A new public comment period, so a chance to weigh in.
When is that?
I just got the email this morning.
They're virtual meetings, like 3 of them, where you have to go online, and then there's going to be one at Sunset Park, a public meeting. I think it's June 13th. Sunset Elementary School, I'm sorry, I meant to say elementary school. So I got it this morning, but I may be getting it from Coastal Federation, because they're very interested in this.
Okay.
So, anyway.
Is UNCW planning to provide this information to the Corps of Engineers independently, or are you relying on citizens like us?
That's a good question. I don't have any plans currently myself, but it's probably a good idea. Yeah. Well, let me hold on just a little bit more because I know for the recording that the Q&A can be hard. So let me get through the last couple slides here and then we can do comments and questions. Okay, so then also, as I mentioned, some of these trees are very old. Old growth trees do offer different ecosystem services than young trees, right? So it's hundreds of years of growth, potentially lost. Again, one of the trees was over 800, but 12 trees were over 200. And this was with a limited amount of coring. So a lot of the trees are older for sure, especially the big ones that are completely hollow on the inside. We can't date with the tree ring methods. So these older trees are more vulnerable is what we expect. That's why the older trees have already been lost in the saltier site. And then thirdly, that stressed trees are more vulnerable to climate change and climate events. So we see stronger relationships between growth and climate when trees are stressed. So the saltwater can be kind of part of this larger story where now the trees are less resilient to climate change potentially. So other implications as well, but I think those are the big ones that I have here. I want to thank a number of different collaborators, so a big team of people who came out on Smith Creek on various days, a lot of funding that supported this research, and definitely my colleagues. Like I said, some of them know more about parts of the research than I do, and I can share those contacts as well. So Kendra was the thesis student, and Andrea Hawks is the sediment foram guru at UNCW. Okay, well, thank you, and I'd love to hear questions and comments.